to be introducing Dr. Andrew Hanson, who has worked for more than 30 years on plant and microbial metabolic biochemistry and engineering, mainly related to primary pathways. So he actually started his career in industry in the UK and then moved to the MSU DOE Plant Research Laboratory at Michigan State University. He then moved to the University of Montreal and then finally ended up in the University of Florida, where he is today. His research portfolio includes crop stress metabolism, amino acid and sulfur metabolism, B vitamin metabolism, metabolic damage and repair pathways. And most recently, he's been working on synthetic biology and metabolic engineering. His research is guided by the aim of utility to agriculture and bioeconomy. And I think I want to point out today that he's been really a leading advocate for progress and development of plant synthetic biology. So it's really a privilege to have him come talk today. So Andrew, please take it away.
Well, thank you for the introduction. Let me share my screen. And thank you very much for the invitation to come to Illinois, which I haven't been to physically in a long time. It was great to meet with some of you this morning, and I look forward to further meetings this afternoon. So I really appreciate the invitation, even though it's a virtual one. This is the formal title of my talk today, Extending Enzyme Lifetime to Increase Net Carbon Fixation. But an informal and punchier one would be carbon demand destruction. And demand destruction is an economics term for reducing the demand for a commodity. And this is going to be clearer what I mean when I talk later as to why I'm thinking it's destruction. Of course, I come from the University of Florida Horticultural Sciences Department. The work I'm going to talk about was done principally by two great postdoctorals, Donato Garcia, who's moved back now to his native Mexico, where he has a synthetic biology position with the Tecnológico de Monterrey. And he was taken over from brilliantly by Kristen van Gelder, who's from Canada, who's been carrying it forward for the last year. And it was begun by Jaya Joshi, another postdoctoral who's moved on to the Biofoundry in Montreal with Vincent Martin. The directed evolution work I'll talk about was very much dependent on Chang Liu, who invented the system we use. Orthorep, he's been a tremendously supportive colleague. And some of the science behind it was done with Harvey Miller, who's pioneered proteome-wide protein turnover measurements in plants, and with Jeff Amthor, who is, of course, a crop modeler and a respiration expert, and they've been really instrumental too in the thinking behind this. And I have to thank the National Science Foundation and even more the Department of Energy for funding this work. Here's the flow of ideas in what I'll say. I'm going to begin with a brief review of carbon budgets, especially of respiratory carbon loss, its magnitude. I'll show you how enzyme turnover drives pretty massive carbon loss from plants. I'll then talk about how enzymes have different turnover rates, different lifespans, and how we can potentially increase lifespan of enzymes by directed evolution. And finally, I'll report progress on a test case, a pretty extreme test case, the Thi4-thiazole synthase, and go on briefly to contemplate extending this approach to other enzymes. Okay, let's begin with carbon budgets. For every 100 carbon units fixed by photosynthesis, net of photorespiration, so we've already fixed it, it's after photorespiration is done what it's going to do. For every carbon units, this is audience participation. About how many are lost during the whole life of a crop? Can I have some estimates, please? from the audience? For every 100 carbons you fix, how many of them do you lose by respiration? Not in the leaf during the day, over the whole life of the crop as a whole organism. No estimates?
They're in the chat.
I can't see the chat.
Well,
I've deliberately, at the direction of your IT person, I've disabled the thing that enables me to see the chat.
I can read them out. So we have 40 from Steve Long and Don Ott. We have 30 from myself and Yuan. We have also 42, 60, and 60.
Oh, this is perfect. You're the best. You're the first audience I've ever asked who got the answer right. I mean, grosso modo 50, but it's somewhere in the range 30 to 60, depending on the crop and the way it's estimated. So this is information from a classical article by Roger Gifford a long time ago for an annual grain crop that, you know, your total pie there is our 100 units of gross carbon fixation. That's the whole pie. Half the pie, 50, then is lost over the life of the crop by respiration. Of that 50, about 25 go to fuel so-called maintenance processes, of which more later. But what I want you to note for now is that maintenance carbon loss is roughly of the same magnitude of the amount of carbon we harvest in yield, in harvested parts. So the concept is, if you could drive down maintenance carbon losses, you would have more carbon that you could put into harvested yield. So the concept is basically changing the slices of the pie. That's a very different approach to the current synthetic biology, metabolic engineering to bioproductivity, which, you know, been so successfully explored in the group here, the Right Project and related work. It's basically to grow the whole pie. So we're thinking that not just growing the whole pie, but altering the size of the slices is another thing, and that they can be combined. They're not mutually exclusive. You can do both, and I think they're potentially more powerful when combined than either alone. Going back to the 25 that are maintenance processes there, the biggest item of that, half or perhaps more, is the cost of protein turnover. And it's that massive cost of protein turnover, it's that makes abundant enzymes that turn over very fast prime targets for improving in terms of slowing their turnover. Well, as we've said, some enzymes turn over fast, some don't. They have different lifespans. And I want to introduce you to a new metric for this, which is another way of looking at protein turnover or enzyme turnover, which is not just half-life of the protein. It's what we call CCR, catalytic cycles till replacement. And this thinking was developed with Harvey Miller. It's the average number of catalytic cycles that an enzyme mediates before it dies. In other words, it's before it's degraded. Mathematically, it's the in vivo catalytic turnover rate, which you either measure from flux measurements or you estimate from metabolic modeling constrained, which is constrained within limits to be realistic, and the protein turnover rate, which Harvey Miller can now measure on a semi-proteome basis. So that's how you estimate it mathematically. For those of you familiar with the protein and catalyst engineering, this is the in vivo equivalent. CCR is the in vivo equivalent of total turnover number, which is for enzymes ex vivo. A little bit more audience participation now. Let's see how well you do on this one. what would you say the median plant enzyme CCR value is? In other words, what's an average plant enzyme? How many times does it do its reaction before it dies? The average one. Again, Steve, if you can harvest the estimates and let me know.
Yeah. I took a rough stab at 20. Now we'll see what else. 400, 200.
20, 400.
200, 100. Steve Long says Rubisco counts as 25% within 2 days. So yeah, well, there's a range there.
Okay, well, if it was only a couple of hundred, the cost of replacing turnover would be so fast that plants would actually shrink, not grow. In other words, it's more than their carbon income. The median value is something like 400,000, nearly 500,000, but there's an absolutely huge range. It's over 7 orders of magnitude. And I'm going to show you that now. What we're looking at here is a cumulative distribution curve for CCR values on a log scale from 10 to the 0, which is 1, up to 100 million. The green line is the plants. So you can see you get the 50% mark, the median value at about 400,000. For bacteria represented by Lactococcus lactis or yeast, your values are an order of magnitude less, and there's somewhat less variation too. So plants spread over a huge range with, I just want you to notice for now that there are some that have very low values, indeed, including a value of 1. But we're going to come back to that. So we think that it should be possible to increase this CCR value and lifespan, informally put, by directed evolution. And before talking about it in detail, I should recapitulate briefly what directed evolution is for those who may not be so familiar with it. First and foremost, of course, it's one of synthetic biology's most iconic achievements, and it was the subject of the Nobel Prize in Chemistry in 2018, awarded jointly to Frances Arnold and 2 others for her work in directed evolution of enzymes. That was classical directed evolution, which consists of repeated rounds of mutagenesis and selection towards a new activity of an enzyme or an improved activity of an existing one. In diagrammatic form, it's this. You start with a gene of interest that you hypermutate in vitro, for example, by error-prone PCR, that's the little X's. You then transform that library of mutants into host cells, where the host cells express the enzyme. You then either screen or, in rarer cases, directly select for the phenotype that you want. Isolate the DNA from the ones you want, subject it to another round of hypermutation, transformation, screening, and so on until you arrive at the end. What I want you to notice there is the color of the boxes, black or gray, for the amount of researcher involvement. The expression step, and when you can do it, selection are passive. There's no human activity needed. All the black ones are active. This involves human labor, quite a lot of it, high skill. That's classical directed evolution. The sort of next gen version is in vivo continuous directed evolution, where the hypermutation and amplification steps occur in vivo, gray color, passive, no active involvement by the investigator. The expression also involves no input directly from the researcher. The selection happens in the cells in vivo, and the selected cells that do better go back hypermutated in vivo, and so you go around with nothing black here. All you're doing basically is subculturing the cells until you've selected them as far as you can. So it's massively less labor requiring, and it gives you a big advantage of what Chang Liu calls depth and scale. Depth means that you can explore much longer mutational walks through sequence space, because you can pick up a lot more mutations here, and you can do it to scale in multiple populations simultaneously, which really you can't when you have to do it manually. So it's hugely more powerful. What it requires, though, is that you're able to couple the activity of the gene of interest, your target, to growth so that you can select. That's the constraint. So how do we think we can apply this to crops, specifically to a carbon budget? Well, in schematic form, it's this. Here's our continuous directed evolution cycle in a yeast or an E. coli platform going round and round, as I just showed you. Here's our plant. The notion is that you can take a wild type plant gene, enter it into this cycle, selecting it for what you want, and then put the improved gene back into a plant using gene editing to introduce the specific mutations that you've selected for in the microbial platform. And that would, directed evolution experiments, typically half a dozen mutations gives you a lot of what you want. So it's within the range of the capabilities of gene editing already to do this kind of manipulation. A second way you can use it, and that's the way we're going to talk about in a minute, is you can take a microbial gene and basically plantize it, make it work better in plants, and then put the improved gene into plants. To return to this graph of enzyme CCR values, remember we talked about some enzymes having very low values in plants, including one, a single turnover, and the enzyme that has that value is Thi4, the thiazole synthase. That's the one I'm going to talk about evolving from now on. I need to make clear that there's no in vitro assay for this enzyme, which is unfortunate because it means you can't measure what you've done by directed evolution by looking at the catalytic features of it. You can only assay it in vivo, which is a limitation, but it's such an important target, we're prepared to deal with that limitation. Plant Thi4s, like yeast Thi4s, are suicide enzymes, hence the CCR value of one reaction and it's dead. That's because Thi4s synthesize the thiazole moiety of thiamine from glycine NAD and a sulfur atom, which in the case of plant and yeast Thi4s comes from a cysteine residue in the active site of the protein, which is used to donate the sulfur for the thiazole ring. In the process, it's converted to dehydroalanine. It cannot be repaired and restored. So the only way you can get your Thi4 back is to degrade it and completely remake it. So to make one molecule of this, you have to blow a whole 300 residue protein, take it apart, put it back together again. Very expensive. Thi4 turnover cost, depending on organism and conditions, rate probably between 50 and 300 micropoles of ATP per gram dry weight per day, which translates into something like to at the very most 12% of the total maintenance respiration. So a large fraction of maintenance is actually going to this single protein, which is the fastest turning over protein in barley and Arabidopsis, by the way. If you settle on a kind of middling figure from that range, of 5% and say, well, suppose we could replace this suicide enzyme with an enzyme that had a lot longer life, say it did 100 turnovers instead of 1, then obviously we cut its turnover cost by a factor of 100, which makes it negligible. Suppose we could in that way reduce maintenance respiration by a middle value in this range, then crop modeling done with Jeff Amthor showed that for an annual crop with a 150-day growing season, that's your wild type biomass accumulation over the season. That's your notional reduced respiration plant, which has a more efficient Thi4 that actually does 100 reactions instead of 1. And your biomass gain is about 4%, which is quite significant and worth having in itself. But of course, this is only one enzyme, and there are others that could potentially be pyramided with it. But let's just focus on Thi4. Fortunately, not all Thi4s in nature are suicide enzymes. There are some prokaryotic ones that are not suicidal. Instead of plundering a cysteine in the active site for the sulfur atom of the thiazole, they use free sulfide. But notice there's an asterisk on that, not suicidal. There's a but, because these prokaryote Thi4s are from pretty well anaerobic organisms that work with little or no oxygen. They typically have very high sulfide concentration, so there's a lot of this substrate. In some cases, millimolar levels, which is of course more than enough to poison a plant or just about anything else on the planet. And they are very poor catalysts. You can't really assay them, but the best estimates of Kcat that we have are about one turnover per hour, which compares very unfavorably with the median value of 1 per second. So this is why we want to try to use continuous directed evolution to improve the oxygen tolerance of these enzymes, to adapt them to low sulfide, and hopefully to increase the Kcat. And you'll notice that those top two are really adapting the Thi4s to plant-like conditions, in other words, plantizing them. Now that's where we get our demand destruction. If we could plantize these catalytic Thi4s, replace the suicidal ones, we would have destroyed a significant fraction of maintenance energy demand. That's the strategy. Tactically, we're choosing to do this in 2 steps. In other words, we're going after oxygen tolerance first, sulfide concentration will follow. That's because you can't apply too much selection to start with, because the enzymes you're working with just can't do very much. It's better to do it. We found empirically it's better to do it, not to try to make them do everything at once. The system we've chosen to use is yeast orthorep, which is Chang Liu's very powerful system. In this system, an error-prone DNA polymerase specifically targets or hypermutates a target enzyme, in our case Thi4, which is on a linear cytoplasmic plasmid called P1. So what we've got here is an error-prone polymerase born on a nuclear plasmid in this case, specifically replicating just P1, which has the target gene in mauve here spliced into it. P2 is a kind of infrastructure plasmid. It's a wild-type plasmid. We don't engineer that. It has to be there. The targets we chose to use in this study are both from anaerobes. One is MH-Thi4 is from a leech gut organism. The other one, SF-Thi4, is from a marine mud organism. In a little more detail, the function of this Thi4 target is coupled to growth very easily, because we just delete Thi4 from the platform yeast strain, so it can't do that reaction. It needs a one on a plasmid. So again, in a little more detail, there's our error-prone DNA polymerase. It's the version called 611 on the nuclear plasmid. Here's our target Thy4 on P1. I emphasize that the polymerase here is absolutely specific to P1. It does not act on nuclear or mitochondrial DNA, so you don't have off-target mutations. We're doing this in a background strain, which is deleted for Thy4. It's also deleted for an enzyme MET15, which will elevate its internal sulfide concentration. So we're doing this one thing at a time with an abnormally high sulfide concentration in the strain we're using. The selection strategy we used, the simplest and the one we made the most use of, was what we call just cold turkey. We put it straight into a medium with no supplemental thiamine and keep passaging it, transferring it between passages for up to 21 times. Screen the mutations, validate them, which I'll talk about later. We did use 2 other selection schemes that I can talk about in questions at the end. Here are the outcomes. In 6 of the populations, we started to evolve. Well, we've started to evolve a lot more than 6, but 6 of them, growth improved progressively over time between passages. So here we're looking in the open triangles at passage 1. Growth started out at about 4 days, reached about an OD of 4. Passage 2, we were already doing a little better. By the time you get to passage 6, we've done a lot better. If you keep that in your mind and then look at the same data, basically, where the purple line is the improved population, the orange line is the parental population we started with, the wild type Thy4. This is the selected one, so we've moved this much. What I want you to notice is that we've moved to us being about as good as yeast Thy4 is itself, the control. That's really the upper bound, excuse me, of the selection window. We can't do much better than that. And of course, we're growing better than the empty vector. When we get to this point of populations that grow a lot better, excuse me, we reclone the genes into fresh plasmid, put them back into fresh yeast to validate that we're looking at a phenotype, a growth phenotype, which is conferred by the ORF itself that we've mutated and not by off-target mutations. And all these 6 that grew better, they did indeed test out that when we reclone them and retransform them into fresh cells, they perform as before. So we're not talking about cheaters here. When we looked at the Thy4s that were improved, they all had 1 or 2 non-synonymous mutations. In other words, mutations that changed the amino acid sequence in the ORF. Here's a summary. The yellow ones, out of a total of rather more than double the number of yellow ones, those are the ones that had 1 or 2 non-synonymous mutations. That's an example of a single synonymous mutation. That's an example. Oops. That's the example of 2 mutations. What we're looking at here is mostly MH Thy4 with a few SF Thy4s, and these are different selection strategies. The strategy 1, the main one that I talked about, cold turkey, and these are separate populations, independently evolving. Interestingly, in 3 of the improved populations, or 3 of the mutations, the same ones showed up in independent populations. That's an example. Y122C showed up in 2 independent populations. V124A showed up in 3, 1 here, 1 here, and 1 here. and D168G also showed up in 3. So what we're talking about here is convergent evolution, that the system is stumbling across the same mutations over and over again in independent populations, in independent, with different selection strategies too. I now want to talk about rationalizing outcomes as best we can as to whether what we've achieved is actually oxygen tolerance or whether it's some other feature of what it takes to make these things work well in yeast. And of course, we're going to get some of those. We're going to get some things that are not to do with oxygen tolerance. They are to do more with what you could call yeastizing. But this one in particular, this is the one we are most confident about, is explained by oxygen tolerance. And here's the evidence for that. This is V124A, this one, which occurred in 3 separate populations, if you remember. And we looked at the natural occurrence in sequences in GenBank, of which there are many, of a V or an A at position 124. Now, V is by far the dominant form. It's about 90% of sequences, and we're talking about hundreds of sequences here. A is a minority form, but it's about 10%. It's definitely there. What was interesting was when we looked at the species and their ecology with the V or the A and at their genomes. Here are 20 typical V species, and here are 10 typical A species. Look first at the first column here, the one that's labeled oxygen. This is a sort of qualitative grayscale representation of the oxygen concentration likely to prevail in the habitat of these organisms as judged from literature on their original isolation and physiology, or from the notes in metagenome projects, with white being no oxygen and black being full air. Well, you can see there's no black here, so all of these are more or less hypoxic or anaerobic. What I want you to notice is the V enzymes, most of them are really strict anaerobes. There's really no evidence that their environment, their habitat typically contains oxygen. There's a couple of exceptions here. But look at the A124s. All of these are in environments in which there's definitely some oxygen at some time and maybe all the time. That's very firmly backed up by looking at the oxygen metabolism genes in genomes, because here we're talking about oxygen defense genes, catalase and superoxide dismutase. Pretty well everything has those. We're looking at anaerobic or aerobic ribonucleotide reductases, and by and large, most of them have both, like E. coli does. It's the ones in the middle, the cytochrome oxidases, that I want you to most look at. Those two, COX and CCO, are more or less high oxygen requiring or high oxygen adapted terminal oxidases. In other words, they have, COX has a really pretty low affinity for oxygen. CCO is higher, but it's not as low as CID. And these relatively high oxygen requiring terminal oxidases, these anaerobic organisms don't have either of them by and large. In contrast, the A-containing, or organisms with an A-containing Thi4 have, by and large, have both of them. So that's the indirect evidence for oxygen tolerance being conferred by V124A. Unfortunately, as I said, we cannot simply assay these enzymes with and without oxygen to test that directly. Well, here are some take-homes from this exercise. The first is that we can definitely improve catalytic Thi4s for function in relatively plant-like conditions in yeast. That can be done. at least in some cases of the mutations, are likely due to improved oxygen tolerance. We can say also that this improvement is easily achieved by various single mutations. We've got a half a dozen single mutations that will take you pretty much to being as good as the yeast control. Now, that's both a minus in the short term and a plus in the long term. It's a minus in the short term. It means that we've reached the top of the selection window. We can't go any further unless we change the expression level or in some other way alter the selection window. And again, in the questions, I can talk about how we can do that. It's not easy to alter the expression level in OrthoRep. It doesn't have an inducible promoter, for example, but there are ways to do it. And so that's what we now need to do. We need to dial down the expression of the enzyme so that we can shift the selection window. But in the long term, it's an upside that you can get there with single mutations, because that means there's an enormous amount of sequence space represented by double, triple, and further mutants left to explore. And it would honestly amaze me if we can't do better than we already did, because it was just so easy to get things that were better with single mutations. Technically, and as a vote of confidence in Chang Liu's OrthoRep, it's durable, which means you can run it forever. It doesn't stop mutating. And it's not plagued by off-target mutations, which the other E. coli systems definitely are, and we can talk about that in questions. We did try 2 other systems before we decided OrthoRep was the one we were going to use. As I said, it's durable because we've carried our cells for more than 100 generations, and they're still hypermutating at the end of that. And it's on target because, as I told you, all the things that we found were improved populations. We found every one of them was attributable to an ORF mutation, because when you sequence the ORF, you found a mutation, you put that mutant ORF back into fresh plasmid and fresh cells, and it had the phenotype. That is not the case with the other systems we tried. You noticed on my slide of mutations that we recovered that there were quite a lot that were not beneficial. Those were mostly neutral. There were a few negative ones in there. We are pretty sure that's because once you get in a population of yeast an improved thiazole producer, in other words, a Thi4 that works somewhat better, yeast releases up to half the thymine it makes into the medium. That's a classically known fact. So your improved thiazole producers in a population can easily cross-feed cheaters. So as your populations improve at making thiazole and thiamine, which is for which the thiazole required, they get better and better at supporting cross-feeding cheaters who accompany them. And of course, because it's a really, really hypermutating system, those cheaters can pick up, certainly pick up mutual mutations. They can even pick up negative ones because they don't need the thiamine to make them, they don't need to make it themselves. They can just pick it up from the medium that somebody else has made. This, by the way, may be a special problem with thiamine, because not many products of yeast metabolism, not many of them are 50% released to the medium. So this may not afflict other kinds of selection regimes as much, but for thiamine it certainly does. So I've shown you that we can go with microbial enzymes from a catalytic cycle of 1 to something more than 1. The next challenge for the future is to go from enzymes in plants that do several cycles to enzymes that do many. In other words, on our green graph of cumulative distribution of CCR values, we can start working on things that are down here in this range, not just the ones that the single one that's at 10 to the 0. Now, we have good hope that that's possible, and this is the logic behind that hope. If you take the Arabidopsis CCR distribution, the population of enzymes, and break them down into those with what we're calling no risk, low risk, or high risk, you find that the ones that we're denoting medium and high risk, and I'll explain what that is in a minute, those are the ones that have the lowest CCRs. In fact, there's practically no low risk and no risk enzymes that are below about 10 to the 5th. There's a few, but even none below 10 to the 4th. So all the short-lived enzymes are what we call medium or high risk. Well, what does that mean? It means that they have risk factors, notably two different kinds. The commonest one is that they are working with chemically very reactive substrates and products, like reactive carbonyls would be a good example, which can readily attack amino groups or thiol groups and form covalent derivatives. In other words, post-translational modifications, which, if they're somewhere near the active site, will inactivate the enzyme. The other kind of risk factor is dangerous mechanisms. And by that, I mean things like radical mechanisms, which are difficult to control, can misfire and damage an active site residue, irreparably, inactivating the enzyme. So you've got this, 1 to 2 order of magnitude shorter median CCR value for the high-risk enzymes, and this great preponderance down here of the short life ones carry these risk factors. So what happens, as I said, is that the reactive substrates or reactive products, let's say reactive carbonyls or radical mechanisms, they hit damage-prone residues somewhere in the active site cleft. We're not talking only about the actual catalytic residues, it's things that are in the neighborhood, bystanders. Many of those damage-prone residues are actually replaceable by less damage-prone residues. And here's an example, not from a plant enzyme, but from a microbial aldolase, which is a very well-known one, that illustrates the point that they're damage-prone and replaceable. Aldolases have a catalytic lysine, which is absolutely invariable. It's part of the catalytic mechanism. This lysine can be cross-linked by the red molecule, which is a chemically formed condensation product of the natural substrate. So it's something that will form in the enzyme active site chemically. You can cross-link between the lysine amino group and the thiol group of a cysteine that's near the active site. And that cross-link is lethal. That inactivates the enzyme, obviously. Either by engineering or by nature, that cysteine doesn't have to be there. You can replace it with something that can't form that link. And when you do, the enzyme becomes completely insensitive to this form of self-inactivation. And that's been done by engineering, protein engineering, but it also occurs in nature. And we're looking here at the relevant region of the sequence of different natural versions of this DOC aldolase. And you can see that the catalytic lysine in green is absolutely invariant, but the cysteine in these three is replaced by another residue, and they are immune from this kind of inactivation. So that's the hope, that we can, by directed evolution, continuous directed evolution, we can harden active sites or active site regions, active site clefts against this kind of collateral damage from the catalytic mechanism or from the substrates and products. We can replace residues that are vulnerable by ones which are less vulnerable. If you want to know a little bit more about the concept of CCR and this idea of hardening against self-inactivation, it's developed in this paper last year in PNAS with Harvey Miller and me kind of bracketing the thing. It was very much a joint effort. If you want to know more about AuthorRep, the wonderful system from Chang Liu, we wrote as a breakthrough technology a kind of user's guide to using it for improving plant enzyme or enzymes for plant applications. And we were privileged to have Chang Liu as one of our co-authors. We do mention briefly in this some of our negative experiences with other systems, and there's another paper which I can refer you to that says a bit more about that. I want to close with putting on a different hat. That's my hat as an associate editor of plant physiology, specifically appointed to handle synthetic biology manuscripts, which we've had very few of so far. That's because we've been building up our editorial team. We've got a second associate editor now, that's Nick Smirnoff in the UK, a real veteran metabolic engineer and metabolic biochemist, really, really deep and good. And 3 wonderful new synthetic biology monitoring editors, Danny Ducat in Michigan State, Alistair McCormack in Edinburgh, and Matthias Zurbriggen in Germany. And so we're very much open for synthetic biology business. And I want to emphasize that we're quite Catholic in our tastes, by which that means universal. The definition of what's in the scope is that photosynthetic organism, synthetic biology. So it doesn't have to be a higher plant. It doesn't have to be a microalgae, although both of those are welcome, of course. It can be a cyanobacterium. It can be an artificial photosynthetic organism. So please think about plant physiology with an impact factor well above 8 for plant-related synthetic biology business for your next submission. And philosophically, we all think, the editors, including the editor-in-chief, that synthetic biology is too important to be partitioned off into specialist journals. It should join the mainstream, not just physiology, ultimately plant improvement. And this is our effort to make that happen. So with that, I'll close and come back for questions.
Great, thank you so much, Andrew. That was really inspiring. So for questions, people can either raise your hand and then I can call you to unmute, or you can put your questions in the chat and I'll read them out. So first hand was Angad. You want to?
Hi, thanks. Thanks, Stephen. Great talk. So I was curious about these set of proteins with low CCRs, right? You had a list of what, 50 to 100 proteins, if I understand correctly.
Well, we looked at a total of something over 100.
Right.
It wasn't as many as we'd like. It was limited by what we could calculate fluxes for and what we had protein turnover data on. So
right. So my question is, what fraction of these proteins or enzymes you have are going to be compatible with
they're all
the
enzymes.
yeast selection? Okay, so what fraction of these enzymes then are going to be compatible with the yeast selection system?
Many of them, because they're primary metabolic. I don't have a number, but we sort of deliberately focused on soluble enzymes because those are the ones that we could evolve, and abundant enzymes because that's the ones we could get protein turnover measurements for. So when you think about what's an abundant soluble enzyme, well, most of them are going to be primary metabolism enzymes. So it's a really good question. Thank you. The enzymes that we did our CCR values for are hugely enriched in soluble primary metabolism enzymes. So because of that, a lot of them are eligible for creating a yeast mutant that they could complement.
Thanks.
Steve Long's raised his hand.
Can you tell us a few? I mean, in those low CCR enzymes, are there a few that will be well known to us?
Oh, absolutely. Maybe I can let me fish around a little bit, and I'll share my screen, because I have a summary of that, or a short summary of the ones we think are the best targets. Go back to screen sharing, Steve.
Yeah. Do you have it? Yeah. Okay. Cool. We can see that.
Yeah. Hopefully you can see this. Here are a half dozen that we picked because they meet a number of criteria. Their CCR value is at least 10-fold less than the medium, in a lot of cases much less than that. They have a crystal structure. They have been either shown to complement E. coli and yeast, or can be highly confidently predicted to do that from the fact that other enzymes, other heterologous enzymes of the same kind can do it. And also, bearing in mind how we've been bitten by thiazole synthase, Thi4, that we can't assay, do they have an assay that you can use to biochemically characterize what you've done? So I think you'll recognize every one of those.
I have a question for you, Andrew. This is kind of related to this. I thought it was really interesting to see that the CCR values for Arabidopsis were shifted relative to both the bacteria and these to the longer phase. Is there a correlation between lifespan of the organism and how you see the CCR? So, like, for example, in your Thi4, you could get some information about oxygen sensitive versus anaerobic conditions. Like, if you did something similar with, like, looking at species with very long lifestyles, will you start to see, I don't know, like in kind of those changes happening in the protein level as well?
No, clearly there's a connotation of a lifestyle association. One thing to note, we were talking about this earlier today, but one thing to note is that the plant fluxes on which CCR in vivo fluxes are calculated on a daily basis, whereas of course the flux is actually a lot higher than that during part of the day. So it could be that they just run with more than they need, because if they were running the whole day at the same flux, they need half as much enzyme, or even a third if it's sort of talking 8 hours, 16-hour day, night, dark. So there is a factor in there related to the fact that yeast and E. coli, at least the way these measurements are made, are being grown in absolutely constant conditions, and so the fluxes are invariant pretty much, whereas the plants are being grown where the fluxes are going from high to nothing in quite a lot of the enzymes, even the ones that are not photosynthetic carbon related, they're still going all over the place. So that's a partial qualifier, but even when you take that factor out, you're still left with a lot of stuff that's different. I mean, I'd rather not speculate, at least in public, but I mean, it's a question. It seems to be real. I mean, we can definitely measure the protein turnover rates. Harvey Miller has done that, and they're sensible. We can only estimate the flux rates, but we're not doing modeling. We're doing a Penning de Vries stoichiometry approach, and they just can't be far wrong. Okay, they could be wrong by a factor of 2 or 3, but not by a factor of 100. So it's pretty certain that this difference, and the same goes for the microbes. I mean, there, the fluxes are much more firmly anchored in measurements, although they are still modeled. They're model fluxes, but they're really good models. And they can't be far wrong. So, yeah, the difference is real. We can talk about it privately, what it might be about, but I don't want to be recorded on this.
Okay, we have time for one last question, so don't always go as hand raised.
Yes, I was just curious, Andrew, can you comment, or do you know, so you have this very large span of CCR values. Can you say whether that's driven more by protein lifetime versus change in catalytic turnover rates?
I can, and its turnover rates are a very large part of the term.
Cool. All right, well, thank you very much, Andrew. I realize we're up to the hour, so if everyone can join me in thanking him again, and then thanks for a great seminar and like interesting discussion session, and yeah, we really appreciate it.
Well, thank you again for the invitation. It's great to be there virtually. I guess I now switch to another link and talk to Don. Is that